Okay, so hello everyone, and thank you for joining us today for the webinar organized by the OTS. And today we're going to be presenting to you our speakers who are going to be talking to us about their work that has led to them winning a poster winner showcase during the OTS annual meeting this year. I am Hassan, and I'm going to be co-hosting this session with Ronald. And just a reminder to the attendees, if you have any questions, please put them in the chat box or through the Q&A, and we'll be taking care of them after each presenter. For today, we have the presenters are Zexiang Chen, Hallie Barber, Samantha Johnson, and Catherine Monopoly. And we're going to be starting with Zexiang Chen, who's going to be talking to us about 5' modifications and how they improve the potency and efficacy of DNA donors for precision genome editing. So Zexiang, if you would like to start, you can go ahead.
Thank you, Hassan.
Perfect. We can see your screen.
Okay, thank you. Okay, thank you. Thank you the kind introduction from Hassan, and also thank the invitation from OTS. So my name is Zexiang Chen. I'm a graduate student in Eric Sontheimer Lab and Jonathan Watts Lab at the UMass Medical School RNA Therapeutics Institute. Today, I'm going to talk about a story about 5' modification improve potency and efficacy of DNA donors for precise genome editing. As many of you are aware that CRISPR-Cas system, which is originally found in prokaryotes as an adaptive immune system, and now has been re-engineered and repurposed as a powerful genome editing tool in the past few years. And the last year, the Nobel Prize was awarded to Jennifer Doudna and Emma Charpentier for this achievement. So to put it simple, in the most wide used form of a CRISPR-Cas system is here I listed two, which one is the Cas9 nucleus, the other is the Cas12 nucleus. They are relying on a guide RNA molecules that will form the ribonuclear protein complex with the Cas nucleus, and the guide RNA will look and search the genome, find the target, and induce the breaks, double-strand breaks on the genome. And then we basically relying on the endogenous repair pathways to repair and fix these breaks. And unfortunately, we can utilize this to create the desired edits on the genome for both research and therapeutic purpose. The most simple way for using the CRISPR-Cas system so far is to just involving the Cas9 and the guide RNA to introduce double-strand break and repair by end joining pathway and lead to usually a small insertion deletions on the genome, which can be now achieved in very high efficiency in both in most contexts. And now this year also has been achieved in human clinical trials by Intellia. So these can be used by most time for knockout purpose. However, in many other cases, perhaps more importantly, what we want to do is to precisely manipulate the edits on the genome, like install precise point mutations, small indels, or install a gene size fragment into the target loci as we want with our control. And this will most likely relying on the other pathway called homology-directed repair pathway that utilizing a DNA donor molecules that carry on the homology arm along with your desired edits. And this will be these donor molecules will involve in a series of homology-directed repair pathways reactions and luckily, if you have successful HDR events, the desired edits, either small stuff or large stuff, will be installed on your target loci as you want. However, naturally, the HDR pathway is limited and inefficient. The two major reasons are, one is it's cell cycle dependent, so it's required the dividing cells. As you can imagine, most for example, in vivo case, most cells are post-mitotic. They actually don't divide. So that's one limitation. The other is actually, in a lot of cases, HDI is actually disfavored by the cells to repair the breaks. They actually like to use end joining pathways to repair breaks. So HDI is commonly facing the competition for the end joining pathways. And one additional factor are actually, so these two major limitations has been kind of addressed by previous scientists, and they published quite a lot papers trying to address these two. However, the overall results are still unsatisfied. And one of the major reasons is that people often overlook one additional component in HDR pathway here, which is the donor molecules that I will be focused on for today's talk. As you imagine, for successful HDR events, you will need to deliver the correct donor molecules into the correct position at double-strand breaks and in the correct time, so that you can have, that's a kind of like a prerequisite conditions for HDR to happen. So the project is all starting from the initial idea or intuition that the Krishna, my collaborator, have this idea was to, if we can somehow appending a nuclear localization signal onto our donor molecules, that perhaps we can improve the donor delivery into the nucleus to boost the concentration of the available donor molecules in the nucleus for successful HDR. In order to achieve that goal, we first make our donor molecules having this adapter sequence by simply just synthesizing a modified PCR primers and do a regular PCR, so that our PCR donors will contain this adapter sequence. Then we synthesize the nuclear localization signal peptide through a linker to a peptide nucleic acids. And hopefully, and these two peptide nucleic acid sequence and RNA adapter sequence will base pair with each other. Hopefully, they will hybridize enough in the cellular context, and then we can achieve installing the NLS on the donor, so that we hope we can get better nuclear uptake for our donor molecules. So to test this, so we make these donor molecules, and to test these ideas, we first choose a reporter assay called traffic light reporter, which basically in a HEK293 T cells. Our colleague is using a lentivirus to integrate a reporter cassette into the HEK293 cell, which contain a broken GFP linked by a T2A sequence to an auto-frame M-cherry. So when there's no genome editing, the cell won't fluorescence at all. However, if you have NHEJ events happen in the broken region, a percentage, roughly 1/3 of the cells will have the appropriate indels that will fix the M-cherry in frame, which will then light up M-cherry. And on the other hand, if you provide a donor molecules and repair the broken GFP precisely by HDR, this will light up the GFP only, and M-cherry are still out of frame. So by using this assay, we can use flow cytometry to quantify GFP positive cells and M-cherry positive cells as a surrogate for both HDR events and NHEJ events. So with that donor in the hands and this reporter assay in the hands also, we went on and carry our experiments and compare the modified donors versus unmodified donor at a quite dynamic range of the donor dosage. As you can see in here, when we first see the results, it's so striking to us that our modified donor improved quite a lot, almost about 2-fold improvement compared to the unmodified donor. What is more interesting is when we including one of the negative control actually, which is just the RNA tag adapter sequence. There's no PNA nuclear localization signal. We found it also improved HDR at a similar level comparing to the PNA NLS modified donor. at least in this assay we tested. And because of this, we went on to test a couple of different additional flavor, which either just tag only or either just have the RNA adapter sequence at 5' end. They both work at similar range in terms of improving HDR events, which is so exciting to us. And then we want to see whether, we want to make sure, ensure to us that this is not just unique to this special reporter assay. So we went on and characterized our modified donor at endogenous loci by using a GFP donor to tag in the endogenous protein, and then we can use FACS to quantify the GFP positive cells as a surrogate of HDR events. So we tested this in HEC293 T cells at 3 different loci, and at all the 3 loci we tested, the modified donor, either tag only or RNA tag version, they all improving HDR comparing to our modified donor, and between 2 and 5-fold. As expected, genome editing is always loci dependent. And also we want to make sure this is not unique to HEC293 T cells. So we're testing a couple other difficult transfect cell lines in like HFF and true cell lines. Although as I said, it's difficult transfect cell lines, so the overall editing efficiency is low, but still our modified donor perform much better than our modified donor, about 2 to 3-fold improvement. So far we have been testing our modifications on double-strand donors. The other one category of the DNA donor has been extensively using is the single-strand donors in mammalian cells. So we want also to see if our modification can improve the single-strand donor. So to test these ideas, we use a reporter assay called GFP to BFP. So the cells contain a GFP. It will, before HDR events, it will be GFP positive, and after successful HDR events, it will convert GFP to BFP, so we can use flow cytometry to quantify that as an indicative of HDR events. To our surprise, in this, when we're using this short single-strand ODN donor, it's about 66 nucleotide, that the maximal HDR efficiency was not improved. However, the potency was much better when we're using modified donor. So to reach the maximum HDR efficiency, the RNA tag modified donor only need is a 10-fold more potent than the unmodified donors. And because not for these single-strand ODNs, they are small, we can synthesize in-house. So we want to see if we can install additional modification on these SSODN to see if they can further boost the HDR. So such as whether adding stuff at 3' to block will further help, or adding the PS, internal PS linkage will further providing more nucleus resistance. So using the assay I just told you, the GFP to BFP assays at a suboptimal donor dosage, we characterize several different modifications. What we found is that, as you've seen before, when you have 5' modification, it improves HDR. However, when you appending 3' modifications using the same RNA tag, either with or without 5' end modification, the HDR was not improved. And especially in the case when you have both 5' and 3', as you can see, the 3' RNA tag actually prevent the HDR improvement from the 5' modifications. And also, it seems like to us, the simple PS, the several PS internal linkage does not improve HDR in this case. However, this is only a tip of iceberg of stuff we can play around on the single-strand ODN. So we are also currently working on more stuff on this. And then, so the data I've been showing you so far is in the mammalian cells. With these all exciting data, we want to move our donor to test them in the in vivo. And to testing our modified donor, as I mentioned before, since in the in vivo case, most cells are post-mitotic, they don't actually have HDR repair pathway actively running. So we choose to test our modified donor in the germlines of 3 model organisms. The first one we're testing is in the C. elegans, the worms. So my collaborator, Krishna and Greg from Craig Mello Lab, they carry on experiments, design a donor either to repair or to insert a full-length GFP into the worm germline cells, or just repair the broken GFP, like I mentioned, similarly, like I mentioned before in mammalian cells. And this is just the 2 loci they've been testing, and I'm showing here, as you can see here, for both tag modified and RNA tag modified donors, they improve the GFP positive F1s, the progenies of worms, compared to the RNA tag about 2 to 3-fold. And this is just 2 loci I've been showing here. And I can tell you, Krishna has been testing so many other loci in the worms. They all work beautifully. It's kind of, I think, suffice to say, I think he kind of cracked the HDR issue in the C. elegans for making this transgenic C. elegans. And the second organism we tested is the zebrafish. So with the help from Nathan Lawson lab at UMass campus, we designed donors to precisely insert a AVI-tag at the HEY2 locus of the zebrafish zygote. And we compare our tag and RNA tag modified donor to the unmodified donors. And as you can see here, the RNA tag donor strikingly improved the precise knocking reads compared to unmodified donors, although tag donor was not improving compared to unmodified donor for the unknown reason yet. So, and the overall knocking efficiency is low because of the low overall general indel efficiency. So as we expect, if we further boost the total cutting events in the zebrafish zygote, we will get much higher HDR percentage. And the last but not least, the model organism we test is the mice. So with the help from the Jaime lab in UMass campus, so we design and test in the 2 different loci. The first one we choose is the tyrosinase mutation, which for these mice, they carry a mutation on the tyrosinase gene that will cause the mice to lose the pigment in the fur along other phenotype. And by successfully fix the mutation in the tyrosinase genes, we can recover the mice with the firm color and other phenotype. So we design a donor contain the proper conversion along with other 6 silent mutations just to prevent the repetitive cutting from the Cas9 RNP. And the Jaime lab help us with the injection and the downstream mouse assay. And very excitingly, when we see the results, that is when our RNA tag donor improve precise repair of the tyrosinase locus in these albino mice about 2 to 3-fold. And this is just a representative image of the litters from different donor group. And more importantly is, it seems like our modified donor also reduced the mosaicism of the HDR of the mice, suggesting the modified donor may promote HDR happen at early stage than unmodified donor, which is another advantage. So the second locus we test is to the SOX2 locus, where we just simply do a precise insertion of the C-terminal V5 tag at the SOX2 locus, and these don't have the phenotype, so we basically just genotype them mouse litters and compare between RNA tag donor and unmodified donor. And strikingly, there's about 5 to 6-fold improvement when you're using RNA tag donors, reaching about 30% of the mouse progenies having the correct precise insertion, and this is confirmed by the sequencing. So with all these exciting and interesting data in the culture human cells, in 3 model organisms. We want to know why and how the modification improve HDR. And so far we have not a definitive answer yet, but we can have 2 hints in our hands. The first one is we learn from literature that is when you pump or deliver a double-strand DNA donor or whatever DNA molecules into the cells, they will ligate to each other by the ligase in the cells or by other enzyme and form either randomly donor concatemer or some kind of head-to-head, head-to-tail concatemer. And these tandem repeats could also be used for either HDR events or end-join events. However, these won't be your desired edits, as you can imagine. So we hypothesize that maybe our donor modifications can suppress these donor concatemer events. So to test this, we transfect unmodified and modified donor into cells separately and harvest the DNA at different time course and run on the gel and do the traditional Southern blot and to compare between them. So this is the results there as I mentioned. So we're comparing the unmodified donor on the left to the modified donor at right at different time course. And I hope you can appreciate this, that as soon as 1 hour post delivery of the donor into a cell, you're seeing a lot concatemer forming at unmodified donors, where it's not forming at the modified donors. And as the time goes longer, there's more concatemer forming, longer concatemer forming, even reaching a very high molecular weight for the unmodified donor. However, that's not the case for the modified donors, which suggests that the modification may prevent the concatemer formation and provide more available monomer and also reducing these wrongly donor template for desired genome editing. And the second hint we have is, we bear in mind is that because the donor, as I mentioned in the previous slides, the donor can concatenarize with each other, ligate to each other. So it can also actually ligate directly into the genome by end joining events, which is undesired editing outcome. As you can imagine now, if the donor molecule was directly ligated into the double-strand break, it will contain additional homology, which is not what you want. So to test these ideas, whether our modification can suppress direct ligation of donor into double-strand breaks, we cultivate and adapt previously methods called GuideSeq, which are using we developed before for detect off-target of the CRISPR-Cas9 events. Simply, it's just you will using a Cas9 RNP, cut the genome at either on-target site or off-target loci, and along with the Cas9 RNP, you also deliver a double-strand ODN that if there's a break, this will integrate into the break site, and you can prep the library and do the sequencing and quantify the reads. So we changing this assay for our purpose, and we compare along with the Cas9 RNP, we deliver either unmodified donor or the modified donors and compare the reads count, whether our modified donor can substantially reduce the reads count from the sequencing. And as we expected, this is the results that either the total count or at different loci. So for all these cases, for both strands, for sequencing purpose, our modified donors substantially reduce the read count compared to unmodified donor, which suggests they suppress the direct ligation of the donor into the double-strand breaks by using modifications. So to summarize my talk, I hope I convince you that the 5' modification is a very straightforward and simple way to improving the HDR events in cultured human cells and also in the germline of C. elegans fish and mice for making transgenic animal models. And also we have some ideas about why they improve in HDR. And we are currently working on, and we believe actually the modifications I just showed you is only a tipberg of ice. And we think there are much more room we can further optimizing and improve our modifications. So we're currently working on the next generation modifications, and we want to apply them into the primary human T cells for making such as CAR Ts. And we also want to further explore why the modification improve so that it can help us better understand and characterize and design our donors. So hopefully I will have more results to present you in the next time. So thank you very much. And at last, I would like to acknowledge all the people. This project has been a wonderful collaboration between multiple labs on the UMass campus. And without all the people listed here and other people, we cannot achieve all the data I just showed you. And especially I want to mention, acknowledge Krishna, which is the leader and the initiator of this project that spearhead and lead the project and get us to here. So thank you very much for your time. And any question?
Thank you so much, Zexiang. Wonderful talk.
Thank you.
So I'll kick off with one very quick question. Have you tried any other modifications other than 2' O-methyl, or is there a reason that you specifically chose that modification?
So we are working on that now. So that's what I mentioned at last slide. So I hope I can bring more results to you. So that's currently under investigation. And the reason initially we choosing 2' O-methyl is because we don't want to just 2' OH will be degraded, obviously, and we also don't want DNA initially because we may afraid polymerase will read through the DNA. However, that might not be the case. So, and we hope, and we are testing other 2' modifications. So we may find a better one. We're not sure about that. So hopefully I can bring more. Yeah.
Okay, perfect. Thank you.
Thank you.
Any other questions, Ronald?
Yeah, I have a few. So just to mention, I just put it in the chat, but if there are any questions, please put them in the chat so we can read the question and the presenters can answer. So my first question is actually on the cell models. So you showed a whole list of different cell lines. I was wondering, did you ever use iPS cells? Because this might be a very good tool to make isogenic iPS cells.
That's a great question. That's something we want to try always. We haven't tried that because of the technical issues. So, but definitely that's something we want to try that, and we do believe that can help a lot for iPSC cells. And yeah.
And then my follow-up question is, I think Hassan also asked something about the optimization of the of whatever you use. So I was wondering. For example, did you try different homology arms and different sizes of insert and the follow-up question? So for myself, I mainly work on repeat expansion diseases. Mm-hmm. So do you have like if you want to make an isogenic Huntington disease cell line, we need a very big insert. So what's the maximum size of the insert and can you play around with these kind of settings?
So we have been testing different lengths of inserts. So far, the most robust we're testing so far is GFP, and we are planning and designing and planning to test a longer insert, such as for CAR T cells. So I don't have that data yet. But I would assume that will also help in the case of longer insertion than the GFP size, which is about 600 to 700 base pair. That's for the GFP size. So I think I'm not sure if that's a length for your repeated. Yeah.
Okay.
Yeah, it might be in a good range. So the follow-up question and the homology arms. Did you play around with the homology arms?
Yeah. So the homology arm, I think we haven't thoroughly played around, but in the experiment we tested, they have been with a different homology length. For example, in the mouse cell, in the transgenic mouse, we are using only 90 base homology. However, in the cell, in the most cellular work, we're using a longer homology because of the GFP insertion size were longer. So, which is what has been reported before. When you're trying to insert a longer stuff, you can't need longer homology. But that's a very good point that we want to thoroughly characterize the homology length, because the modification may perform, like you said, may perform much better at a certain length, homology length, maybe in a shorter length or maybe in a longer length. Yeah.
All right, perfect. Thank you so much, Zexiang. That was a great talk, and we'll be looking forward for more work from you, hopefully next year.
Yeah, thank you very much.
Okay, so we're gonna move on to our next speaker, which is Hallie Barber from McGill University. And she will be presenting to us about the chemical modifications in the CRISPR-Cas12 system for gene editing. So Hallie, if you wanna share your screen, the floor is yours.
Sorry, I believe I'm still muted. Yes, thank you for the introduction. And let me just move my screen.
We can see your screen and we can hear you perfectly. So
all right. Yes, so thank you, Hassan, for the introduction, and to both you and Ronald for hosting this session. And thanks for OTS for inviting me to talk about my research. My name is Hallie Barber, and I'm a second-year PhD student at McGill University in Dr. Masad Dama's lab. And today I'm going to be talking about a project that we have been working on in collaboration with Dr. Keith Gagnon's lab at Southern Illinois University, having to do with chemical modifications within the CRISPR RNA of the CRISPR-Cas12a system for gene editing. So first, since we just heard about CRISPR, I'm going to just give a brief overview of the specific system. So CRISPR-Cas systems are innate bacterial immune systems that have in recent years been harnessed for gene editing in mammalian cells. So as you can see here, this system is composed of a CRISPR RNA and a CRISPR associated or Cas effector protein. Sorry, I just want to turn on my laser pointer. All right, which can assemble to form a ribonucleoprotein complex. And this complex is often referred to as genetic scissors because it has the ability to induce both site and sequence specific endonucleolytic cleavage of target genomic DNA. And this double-stranded break that results of this cleavage is either repaired by non-homologous end joining, which is often error-prone and leads to knockout of the gene, or it could be repaired by homology-directed repair, in which a DNA template can be knocked in to edit the gene. And the two most well-studied CRISPR-Cas systems are Cas9 and Cas12a. So Cas9 is often derived from Streptococcus pyogenes, while Cas12a is from Acinomenacoccus species. And both are class 2 effector proteins, meaning that they are single multi-domain protein with a bilobe structure, as opposed to a complex of multiple Cas proteins. And Cas9 is categorized as type 2, while Cas12a is type 5. And the main difference between these two systems are that Cas9 is a dual-guided system that is composed of both a CRISPR RNA and a tracer RNA that can be synthetically replicated to be a single guide system that's approximately 99 nucleotides in length and preserves the stem loop structures from the tracer RNA in order to bind to the Cas9 protein, while Cas12a is a simplified single guide system with the 39 nucleotide CRISPR RNA, and it binds to Cas12a through a pseudoknot found at its 5' end. And additionally, Cas9 is known to target T-rich protospacer adjacent motifs or PAM sequences that are found downstream of the cleavage site. And the protein induces blunt double-stranded breaks that are near the PAM site, while Cas12a locates T-rich sequences, T-rich PAM sequences that are upstream of the cleavage site, and it induces staggered double-stranded breaks that are in the PAM distal region, which is important because this contributes to the high specificity, target specificity of the system. All right, so first, Cas12a locates the target gene using a shape and base readout mechanism to scan the genomic DNA for the PAM site on the non-target strand. And once the site is located, the protein initiates unwinding of the double helix to allow for base pairing between the CRISPR RNA and the target strand of the genomic site. And once full complementarity is reached and an R loop structure is formed, the endonucleolytic active site cleaves first the non-target strand at the +18 position, and then the target strand at the +23 position, which results in cleavage products with 5' overhangs. All right, so the most important component of this system and the nucleic acid part is the CRISPR RNA. So this RNA molecule, as I mentioned, has a 5' handle, which has the pseudoknot structure at its 5' end, and it's highly conserved in approximately 19 nucleotides in length across multiple organisms. And then it has at its 3' end a guide region, which is responsible for base pairing with the target DNA. And as you can see by this diagram over here, you have the Cas12a in complex with both the CRISPR RNA and the target DNA. And highlighted below is the structure of the pseudoknot, which has purple highlighted regions, which correspond to 2' hydroxyl contacts. And these are just important hydrogen bonding interactions between the 2' hydroxyl groups of the ribose sugars within the CRISPR RNA and the amino acid side chains of Cas12a, which this is responsible for the correct positioning and association of the ribonucleoprotein complex. So while CRISPR-Cas12a is a highly promising tool for gene editing and therapeutic applications similar to, as you've seen the recent studies of the Cas9 system in clinical trials, however, the drug metabolism and pharmacokinetic properties could be further tuned using nucleic acid modifications in order to stabilize the RNA molecule. So specifically, these modifications can enhance the metabolic and thermal stability of the molecule, as well as its bioavailability. It could reduce its degradation by endogenous nucleases, and as well as the elicited host immune response. And finally, which is critical to the pseudoknot region, it could influence its secondary structure. So often these chemical modifications are found in one of 3 positions, either within the nitrogenous base, the ribose sugar, or the phosphodiester backbone. And throughout all of these studies, we'll be primarily focusing on the ribose sugar here. So in our lab, we used a large toolbox of chemical modifications, and these are example of the nucleotide analogs that contain modifications at the sugar C2' position. So they could be further classified as either RNA-like or DNA-like. So RNA-like analogs have a north or C3' endo sugar pucker conformation and form A-form duplexes. So these nucleotide analogs include 2' fluoro, block nucleic acids, and 2' O-methyl and 2' methoxyethyl, while DNA-like DNA-like analogs have a south or C2' endo sugar pucker conformation and form B-form duplexes. And these include arabinonucleic acids, 2' fluoroarabino, which is also known as FANA, and then 2' amino. So in this slide, I'm just going to overview our original studies having to do with the chemical modification of this pseudoknot region, which was carried out by multiple former group members. And In this first series of modified CRISPR RNAs, we wanted to study the effect of helical preference within the 5' handle. So to accomplish this, we incorporated DNA substitution since, as I previously mentioned, DNA is known to have a B-form helical structure. So in these studies, enhanced green fluorescent protein, or EGFP, was used as a proof of concept target gene, and the CRISPR RNAs were then lipid transfected into human embryonic kidney 293T or HEC293T cells that were expressing both the EGFP gene and the AsCas12a protein. And then loss of fluorescence was used as an indicator of gene knockout. So all of them are recorded in percent EGFP editing. And as you could see from this data, each of the modified sequences resulted in little to no EGFP editing, which led us to conclude that it's the conservation of overall A-form helical preference is important within this 5' handle. And then in our second series, 2' fluoro was used instead of DNA since it has a strong A-form helical preference. So fluoro modifications were incorporated at all of the positions within the 5' handle, except for at the 2' hydroxyl contacts, which are denoted by these red stars at the bottom. And then the fluoro modifications were walked through the contacts to determine whether there were any that were critical to gene editing. And this data shows that loss of terminal contacts, so at the -1, -17, and -18 positions, were not tolerated, while the internal contacts were more tolerant to substitution with 2' fluoro. And interestingly, the sequence SJ14-6, which is shown right here and has the loss of the -6 contact position, it actually had equal editing to the native CRISPR RNA. And this is important because this sequence only contains 6 unmodified RNA nucleotides. So the majority of them have been replaced. And overall, we established that the majority of the hydroxyl contacts within the 5' handle are critical to maintain for sufficient gene editing. However, it is possible to substitute some of them for non-RNA analogs. And then finally, in this third series, we introduced 2' amino into the heavily fluoro-modified sequences. So while, as I showed in the previous slide, 2' amino is a DNA-like analog with B-form preference. It has hydrogen bonding capabilities and therefore could potentially be a sufficient replacement for the hydroxyl contacts, unlike 2' fluoro. So this data shows variable gene editing activity with substitution of the -14 position, right, in the sequence LA-N14-7. It provided the highest activity. So this suggests, and all of the other ones had variable activity. That likely means that 2' amino cannot be used at these positions, but this data overall suggests that RNA and DNA-like nucleotide analogs can be used in combination within this region, and more importantly, that the heavily fluoro-modified sequences can be used to mitigate the negative effects of otherwise beneficial B-form analogs such as 2' amino. All right, so to further probe how modifications within the 5' handle affect the pseudoknot structure, we designed the series to analyze the base pairing region within the stem loop. So which is the base pairing region is right here. So for DNA or DNA-like, in this case, FANA, nucleotides were incorporated into the rightmost base pairing region. So these nucleotides right here. And this was specifically to focus on the canonical base pairing interactions within the stem loop and not to involve the non-canonical base pairing interactions right here, which are responsible for the complete formation of the pseudoknot. And additionally, all of the sequences except for HBA09 retain all of the hydroxyl contacts, and this was just to completely decouple this property from the helical structure. And as I mentioned, FANA was systematically incorporated into this region, so walking this way and then more extensively in the later sequences. And this is because SPANA is known to have an increased binding affinity for RNA, and therefore could potentially stabilize the base pairing regions. And unfortunately, when these sequences were tested in the gene editing assays, they all resulted in little to no gene editing. And we hypothesized that this is this impact on gene editing could possibly be explained by the inability of the pseudoknot structure to form, and therefore for the CRISPR RNA to correctly associate with the Cas12a protein. And therefore we decided to use thermal stability or TM measurements in order to analyze the folding within this region. In this analysis, we only used the truncated 5' handle sequences to not observe any interactions that might have been within the guide region. So so in this graph right here, as you can see, RNA duplexes have lower absorbance than single-stranded RNA, and therefore in the unmodified handle, you see the characteristic sigmoidal melting curve of the RNA as the system is heated up and the base pairing interactions are broken apart. And although with the TM values of each of the modified sequences, they were all lower than the unmodified sequence, we did still observe this characteristic sigmoidal curve as opposed to a flat line, which would indicate that there was no base pairing going on. So this suggests that some secondary structures are still forming, and therefore, although these modification patterns are not viable for gene editing studies, and we will not continue to move forward with these sequences, they still do provide valuable insight into the sensitive nature of the 5' handle. And we can conclude that consecutive B-form nucleotide analogs, similar to the previous studies, preserve the structure too greatly for efficient gene editing, and therefore moving forward, if we choose to continue to modify the 5' handle, we will focus on more A-form nucleotides, or possibly the using the 2' amino sparingly. So using all the information we gathered about the 5' handle, we decided to move on to modifying the guide region, since our goal ultimately is to create a fully modified CRISPR RNA. And similar to the structure-guided approach that we used with the 5' handle, we decided to incorporate 2' fluoro at all of the positions within the guide region, except for at the 2' hydroxyl contacts. And then we walked 2' fluoro through the contact positions to see whether any of these were critical to gene editing. And we're excited to see that each of these sequences resulted in high gene editing, with no one contact particularly important for editing activity. And encouraged by these results, instead of walking one at a time, we walked 2 fluoro modifications at a time through the base pairing, sorry, not the base pairing, the contact positions, and as well as the HBNC sequence was a negative control, since all of the positions, including the contacts, were replaced with 2' fluoro. So we wanted to see whether these contact positions are even important in the guide region. And unsurprisingly, this sequence had very low editing of the EGFP gene. However, it wasn't completely reduced, which is important to note. So that just means that the guide region is less sensitive than, or less sensitive than the 5' handle, and less reliant on these contact positions. And then aside from this sequence, each of the others maintained relatively high editing with more distinct patterns visible. So in particular, the contacts within the first 7 positions of the guide region. So these ones were more sensitive to fluoro substitution than the contacts at the 5' end. And since this region is often referred to as the seed region, since it nucleates the base pairing interactions and the heteroduplex formation between the CRISPR RNA and the target DNA strand, it's possible that this region is more sensitive to modification and loss of the 2' hydroxyl contacts, since these contacts may be important for the Cas12a protein to position the RNA to be able to bind to the target DNA. And moving forward, we want to keep these 2' fluoro modifications since they seem to be well tolerated throughout this region. And we confirmed that it could even tolerate loss of multiple contacts, which was not seen in the 5' handle. So we want to see if, especially in the 3' region, whether these sequences could be replaced with more than two contact positions can be replaced with 2' fluoro, and just to see how extensively we can modify this region. And then at the same time, using different modifications, such as 2' O-methyl or LNA, or possibly 2' arabinose in the seed region to see whether we can bring back these hydrogen bonding interactions and still modify the sequences. So in summary, all of these studies have shown that we can tune the properties of the CRISPR RNA using different nucleic acid chemical modifications. And both the 5' handle and the guide region were successfully modified without compromising the gene editing activity. So we were able to establish different patterns that we could use for modification. And finally, the structure-guided approach can potentially be used to create a fully modified CRISPR RNA with remaining RNA nucleotides stabilized by phosphorothioate linkages. So we saw in data that I did not show that if you replace, if you put phosphorothioate linkages at the 3' end of unmodified RNA, this enhances nucleus stability while also maintaining the hydroxyl contacts, which appear to be important. So these studies were conducted in the pseudonaut region, and we hope to apply this to the guide region as well. And with that, I'd like to thank both the DAMA lab and the Gagnon lab and all of the members for their contributions to this work. And I'll be happy to take any questions.
Thank you so much, Hallie. Beautiful work. So I will kickstart with a very quick question. So when you did the melting temperature experiment, you said that you use it at truncated like 5' pseudonaut. I was wondering if, since FANA has a very high like binding affinity towards RNA, I was wondering if you do it on the full strand, that you would find like any other secondary structures forming. Have you like thought about this, or you think it won't be enough to reach?
Yeah, that's a great question. So originally, when we were synthesizing these sequences, primarily when we were only modifying the pseudonaut, we would just synthesize the pseudonaut, or so this 19-mer individually, and then we would then ligate the 5' handle to the guide region. So we were only synthesizing them in portions, since the guide region remained the same and you can just use the same sequence. So this is why we did the pseudonaut, the TM studies just on the 5' handle region. However, now that we're modifying both regions, it's much simpler to synthesize the entire 39-mer. And going forward, we will be seeing whether there are any complications introduced by base pairing between the 5' handle and the guide region, which there shouldn't be, based on the fact that the pseudonaut can still form and effectively bind to the Cas12a protein, which would not happen if there was base pairing, but it would be interesting to study whether there are overlapping interactions.
Perfect, thank you. Any other questions, Ronald?
Yeah, I have a question. So I think this is all. I think congratulations for this very nice presentation, first of all, and it's very nice work. And I think you very accurately modified all the different positions, and it's very elegant. But my question is, did you also, because this is all done like in a simple cell model, so would this also work in more difficult cell lines or even in animal models? Do you have any data on this? Or
so we do not have any data at the moment, but that is where we're currently moving towards, since we're starting to develop a pattern that can be used to create the fully modified sequence. However, like you mentioned, if this wants to be used in therapeutic applications, it needs to be applicable to human genes. So our current model is just used for EGFP, but we're going to try to change the sequence of the guide region, since that's the great thing about CRISPR systems, is that they could be programmed to different gene targets. And we can use different, both the different human gene targets as well as different cell lines. So right now we're using human embryonic kidney cells, but we're looking into developing it to be more applicable to diverse targets. But currently we do not have that.
Yeah. And do you think that really these motives are specific for every target that you use, or do you think you need to determine motives for every single target that you want to target?
We're hoping that this is applicable to different targets. So since we found that the patterns mostly rely on the helical structure and then the hydroxyl contacts. The hydroxyl contacts are between the CRISPR RNA and the protein, so they should not change based on the gene target. But the helical structure may have an impact based on the target and the accessibility, especially within a genome. So that is why it's important for us to, moving forward, look at whether the same, now that we've established a modification pattern, whether this could be applicable.
Okay, thank you.
Transferable.
So a very good question before we move on. I know that you mentioned that the advantage of using these chemical modifications is also to improve on the stability of the strand. So I wonder if you started looking at like the different modification patterns and if there's any variation in terms of stability between them at specific positions. And like more specifically, I'm just curious, is the 2' amino, like how does it compare, for example, to 2' fluoro in terms of improving stability?
So I don't believe that there's a difference in stability between 2' amino and 2' fluoro, other than the fact that they're both non-canonical nucleic acids. So they aren't recognized by the same host mechanisms for degradation. However, amino is a good choice to use because it still has hydrogen bonding properties. But in terms of the stability, I didn't show the data on these slides, but we did conduct nucleus stability assays in FBS serum, and we're able to see that when, especially with the phosphorylated 5' handle, the unmodified guide region at the time was degraded by nucleases found within the serum, while you can still, these were page gels, and you can still see that there was a band corresponding to the pseudonaut. So this sequence was not degraded by the nucleases, which is important, since that is our main goal, is that this molecule is stable enough to survive entering cells and reaching its target without being degraded.
Perfect. Thank you so much, Halle, and good luck with your future experiments.
Thank you.
So we will be moving to our next speaker, which is Samantha Johnson from the University of Texas Southwestern. And she will be talking to us about scaffolding protein TNRC6 paralogs and how do they impact gene expression and splicing. So Samantha, the floor is yours.
Hi, everybody. I get the slide to move forward. Okay, so hi, my name's Samantha Johnson. I'm really excited to speak to you today about my project, which is the impact of the loss of TNRC6 on RNA interference. So just a quick outline of my talk. I'm going to give a background of RNAi, talk about when TNRC6 is necessary, the functional overlap of Argonaut TNRC6 and TNRC6's involvement in gene splicing. So classical RNA interference sort of goes like this. You have a pre-microRNA in your nucleus that is then exported into the cytoplasm, where a dicer picks it up and forms it into a mature microRNA. It's then loaded into Argonaut, which forms this RISC complex with TNRC6, which is our protein of interest. And then this mature RISC complex goes and targets a mRNA on its 3' UTR, and this is traditionally thought to cause only translational repression. So what we really wanted to do is get a better understanding of RNA interference. So I think everybody generally recognizes the importance of Argonaut and RNAi, but we believe TNRC6 has this potential to be basically the hub of protein interactions for RNAi. And we think by understanding TNRC6 better, we can really elucidate mechanisms of duplex RNA and microRNA and how they can be used as therapeutics. TNRC6 is also really interesting because it's in the nucleus and it could impact the control of splicing and transcription by duplex RNA, which is a exciting new area of therapeutic research. So back to our most popular protein, Argonaut. So there's 4 Argonaut paralogs in humans. You have Argonaut 1, 2, 3, and 4. It's the targeting component of the RISC complex. So it has this ability to bind small RNA, so microRNAs, siRNAs, other things. And then Argonaut 2 has this special cleavage activity where if it has a fully complementary small RNA, basically an siRNA, it's able to cleave a target mRNA. And that's the special thing Argonaut 2 can do. So if you look at proteins that Argonaut 2 interacts with, mass spec basically reveals that Argonaut 2 doesn't interact with a wide variety of proteins. It mostly interacts with other Argonauts, TRBP, and dicer, and then mainly it interacts with TNRC6 paralogs. So you might be asking, what's TNRC6? So it's also known as human GW182 protein. And there's basically 3 paralogs in human and mammalian cells, TNRC6A, B, and C, and it's a scaffolding protein. So what it does is it binds a wide variety of proteins and brings them all together. So it can bind up to 3 Argonaut proteins. So it binds those at this motif 1, motif 2, and the hook with these GW repeats. And it also bridges interactions between these Argonauts and their targets with other proteins like CCR0 and other members of the RISC complex with its silencing domain. And so in the nucleus, TNRC6 is present, and the proteins it interacts with are CCR0 complex, the mediator complex, and histone modifiers, which really indicates that it can be this organizer for RNA-mediated control of transcription, because transcription requires a lot of proteins and a lot of things to sort of get it started. So sort of the functions of Argonaut and TNRC6 that we are really testing and trying to examine more in the cytoplasm are your classical siRNAs, so where you have a fully complementary small RNA in your Argonaut, if it binds to TNRC6, if it's required, is the question we're asking. And then that can basically come and cleave this targeted mRNA. And then the sort of more endogenous form is a microRNA that has this central mismatch. It binds to its target mRNA, and that causes mRNA degradation. So we're really trying to look into TNRC6's involvement in these processes. And then in the nucleus, we're really interested in Argonaut and TNRC6 being able to activate or inhibit gene transcription with a small RNA. And then also we're interested in splicing. Can TNRC6 and Argonaut and a small RNA influence splicing? And is TNRC6 required for that? So in order to study TNRC6 better, we created these TNRC6 knockout cell lines. We have a TNRC6A knockout, a B knockout, a AB double knockout, And then we were actually unable to create a TNRC6C triple knockout cell line. As soon as you try and knock out C and the AB, all the cells die. So we use a pseudo triple knockout where we use a very strong pool of siRNAs to knock down TNRC6C in our double knockout cell line. So now I'm going to be going into when TNRC6 is necessary. So we're asking this question, what RNAi functions require TNRC6 paralogs? So the things we're asking, again, are siRNA knockdown in the cytoplasm, microRNA assays, which takes place in the cytoplasm, and then in the nucleus, we wanted to look at specifically nuclear siRNA knockdown, and then also transcriptional activation. So siRNA knockdown is widely used for a lot of experimental protocols in a lot of various labs. And then there's also on the Therapeutics side, approved drugs that use this siRNA platform. But when you shift to microRNA, we do know that TNRC6 in the past has been shown to be involved in microRNAs. And we also know that there's no microRNA therapeutics that have been approved. So we want to see where TNRC6 is involved. And if we can understand that better, perhaps this could lead to therapeutic developments. So to answer this first question, TNRC6 is not involved in cytoplasmic knockdown by siRNA. So as a negative control, we used an Argonaut-2 knockout cell line. So as you can see here, if we lose Argonaut-2, we don't get any knockdown of ataxin, which is our protein that we are assaying for this. But you do get that knockdown in the wild type, and then in all of our TNRC6 knockdowns, we get really efficient knockdown of ataxin, showing that TNRC6 not really required. So to test the microRNA pathway, we use microRNA 34A. So this is a microRNA that basically induces apoptosis in the cells. It represses SIRT1, which in turn represses P53. So if you remove that repression, you cause apoptosis. So how we measured this was looking at the relative number of live cells. So you can see here in our wild type, we have only about 20% live cells remaining, whereas in our Argonaut-2 knockout, we're basically removing the main Argonaut paralog in the cell line. We have about 80% live cells remaining with our microRNA 34A transfection versus a non-targeting control transfection. So again, Argonaut-2, we have about 80% of cells remaining. And so when we have our TNRC6AB double knockout cells, we actually have about 50% of the live cells remaining, showing that this microRNA assay isn't working as efficiently as it could. And then when we finally knock out TNRC6C, we get about 80% live cells remaining, which is very similar to Argonaut, showing that at least one TNRC6 paralog protein is required for microRNA knockdown to work efficiently. So now we're going to move into our two nuclear functions, which is, again, siRNA knockdown in the nucleus and transcriptional activation. So we found out that TNRC6 is not involved in nuclear knockdown by siRNA. So again, we have Argonaut-2 as our negative control, showing that we don't get good knockdown of mallet1, which is a nuclear long non-coding RNA, but we do get good knockdown in all of our TNRC6 knockout cell lines. And the reason we thought TNRC6 might be involved in nuclear knockdown is just because of the movement to the nucleus. We thought maybe TNRC6 might be required to move a loaded Argonaut in, but evidently it's not. And then the next thing we assayed was transcriptional activation. So the Corey lab has studied this cyclooxygenase 2 transcriptional activation pathway for about 15 years. So it's sort of the best known assay we had to test transcriptional activation. So what happens in this pathway is you have your Argonaut-2, which is binding to a small RNA, which then in turn binds to a non-coding RNA that is transcribed across the cyclooxygenase promoter region. So this binds with TNRC6, possibly, to then activate and bring all the other proteins, which then activates transcription of this cyclooxygenase 2. And then additionally, it activates this chromatin neighbor PLA to G4A, but we don't assay that. So again, we have our assay. So you can see in the wild type, we have about threefold activation of this cyclooxygenase 2 when we transfect our 12NC, which is our activating small RNA, versus our non-targeting SIGL2. So you can see in Argonaut2 and in TNRC6, we don't get this activation of the cyclooxygenase 2 protein, which is really interesting. However, when we get to our TNRC6B, we do see a little bit more activation, which sort of indicates that perhaps it's TNRC6A that is required for this. And then when we get to our sort of pseudo triple knockout, we don't get any more activation. So TNRC6A is the highest expressed paralog in the cell. So it could just be that there's enough TNRC6A remaining to activate this. And the loss of B isn't enough to really impact the population, but since we lose A, TNRC6B is lower expressed, that is enough to impact it. But it's really an interesting result showing that you sort of need most of the proteins to activate. So just to sort of summarize this section, TNRC6 paralogs are required when Argonaut needs additional proteins in order to complete its functions. So transcriptional activation requires recruitment of histone modifiers, RNA polymerase, and a lot of other activating factors. So this is when TNRC6 as a scaffolding protein is really useful. And then again, in the cytoplasm with microRNA-induced translational repression, this requires recruitment of decapping and deadenylation factors and a lot of additional proteins. And TNRC6 is required for that because it brings all those things to this targeted mRNA with its Argonaut. And then conversely, TNRC6 paralogs are not required for the siRNA pathway because Argonaut2 has this cleavage activity without any additional proteins. And once you have a non-protected RNA that's been cleaved in half, the other factors in the cells that degrade RNA take care of it very quickly. So you don't need additional proteins. They'll find it on their own. So the next section I'm going to talk about is sort of this functional overlap of Argonaut and TNRC6. So a question we really had in our lab is how do we find these bona fide endogenous targets? So there's a lot of published microRNA papers that you can find on PubMed, but a lot of them are very difficult to confirm experimentally. They're very particular to one cell type and one condition. So we wanted to use RNA sequencing and Argonaut2 eClip in our knockout cell lines and sequencing them so we can try and sort through and find these endogenous targets of RNAi that are just happening natively in this cell. So Argonaut2 eClip is enhanced cross-linking and immunoprecipitation. It's basically finding out exactly where Argonaut2 is bound in the cell. So you're able to cross-link Argonaut2 to its RNAs, lyse the cell, digest all the RNA, pull out all your Argonaut2 and its protected RNAs, then digest your Argonaut2 proteins, extract your RNA, and then you're able to sequence that and see exactly what RNAs Argonaut2 was bound to, both the small RNA and the transcript, so the targeted mRNA or other things. So our sort of experimental scheme for this is we had a Argonaut2 knockout cell line, which we used as a control for our eClip, as well as our wild type. And we did both cytoplasmic and nuclear fractions separately, so we could understand where Argonaut is binding in the cytoplasm and the nucleus. And we did Argonaut2 eClip with that. And then for RNA sequencing, we had our wild type, our single knockout cell lines, our double knockout cell line, and then our pseudo triple knockout cell line. And then we also had our transfected cell lines, both with our TNRC6C siRNA and then a non-targeting siRNA, just so that way we could compare our pseudo triple knockout with another transfected cell line to eliminate any changes that occur during transfection, because that can cause changes in cells. So just trying to make sure everything is equivocal. So the question we're really trying to answer is what happens to genes in these TNRC6 knockout cell lines? And we predicted since we are losing this protein that's really involved in the repressive pathway, we would have a lot of upregulated genes. And then we were also asking sort of, do the paralogs show any unique changes in these cell lines? Because we're still trying to tease out whether these TNRC6 paralogs have really unique functions that the others can't compensate for. And we predicted there'd be minimal changes in the singles and sort of the most in the pseudo triple knockout cell line, because that's sort of what we had seen in our functional assays. So interestingly enough, we did see that the single knockout cell lines had the least amount of changes. The double and the triple had even more than those. But TNRC6A does have the most significantly changed genes, which does make sense because it is the highest expressed of the paralogs in the cell line. And it may have sort of a little bit of a wider function than A or B, than B or C. And then also interestingly, we saw that we had about equal up and down regulation. And immediately we thought, well, this could be due to sort of more downstream effects, and these aren't genes that are being repressed. So the next thing we wanted to ask is what happens to these transcripts when Argonaut 2 is bound. So just bringing us back to sort of this traditional model of RNAi, we have our TNRC6 in our Argonaut, and that causes gene repression. And then if we remove that, we cause upregulation. So we expected we'd see more upregulation if we were only looking at transcripts where Argonaut 2 was found to be bound. And interestingly, we saw the same amount of up and down regulation again, and this really indicates that there's no simple correlation between this Argonaut occupancy and up or down regulation of that transcript, which we thought sort of opens up this world of RNAi. And this was also seen in Argonaut 2 knockout cell line and other Argonaut cell lines that I'll talk about in just a moment. So sort of the next thing we asked is, do these changes in TNRC6 and Argonaut mirror one another? They're basically protein partners. Argonaut requires TNRC6 for these complex functions. So if we knock one of them out, are they going to mirror one another? So to look at this, we did have these additional TNRC6, these additional Argonaut knockout cell lines. So you have Argonaut 1 and 2 single knockout cell line, Argonaut 1, 2 double, and then Argonaut 1, 2, 3 triple knockout cell line. So we decided to look at the double and triple and just see where there's significant genes overlap that have Argonaut 2 binding clusters. So there's these 67 genes that we with that all have significant changes in all 4 of our cell lines and also have a significant Argonaut 2 binding cluster. So when we look at these genes that we think are going to have endogenous RNAi control, we see that these changes are really mirroring one another for the TNRC6 and the Argonaut knockout cell lines. So the single knockout sort of SIC Argonaut 1 don't have as much congruency with those changes, but the double and the triple show this really great mirroring effect in both the up and down regulation. So this really answers the questions that these knockouts are mirroring one another in terms of gene expression changes, sort of emphasizing that Argonaut and TNRC6 are really close protein binding partners. So the last thing I'll talk about is sort of TNRC6 and gene splicing. So getting back to this last function that I said we wanted to explore is alternative splicing with TNRC6. So we know that TNRC6 is found to be an association with splicing factors in the nucleus. There's 2 papers that were published that showed associations. And so we thought the best way to look for these potential endogenous splicing is to, again, compare the double and triple knockouts and look where Argonaut 2 is bound in the nucleus. So quick splicing review, you have exons and introns. Introns get taken out for a mature mRNA. But you can also have alternative splicing events where an exon can be included or skipped. And those are the specific events that we examined as skipped exon events. So there's actually only 15 events that overlap between all 4 of these cell lines. And then when we layer on where Argonaut 2 is bound, we only have 2 that have Argonaut 2 clusters bound nearby. And by nearby, we just mean within the same intron or exon that's being alternatively spliced. So this is DEPDC1 and EPBL41L2. So the next thing we did is look specifically at the sashimi plots to make sure that what the data was showing us was occurring in terms of the number of reads. So as you can see here, DPDC1 shows an increase in exon skipping. So it's skipping this exon with more and more frequency. And then sort of conveniently, EPBL41 shows the opposite effect where it's including this exon more as we go from wild type to ABSIC. So these are sort of 2 great examples to explore. So we confirmed this with qPCR, so DPTC1 and our AB and our ABC. The qPCR is confirming these changes, and EPBL1 is also confirming. And then we also sort of expanded this list to look at other alternative splicing candidates. And the TNRC6AB cell line did show this alternative splicing and these other 5 genes, showing that we can sort of expand outside of our narrow range and still see these endogenous splicing changes that are lost when you knock out TNRC6AB. And then the last thing we wanted to test is see if we could directly modulate this splicing change by transfecting a double-stranded RNA. So we chose EPBL41, and we transfected, or we sort of designed double-stranded RNAs and transfected to the region where the Argonaut 2 is found to be found. And we showed that we could alternate the splicing in the wild type, but not in the TNRC6AB, emphasizing that for this change, TNRC6AB is required to modulate this splicing. So just to quickly go over my conclusions, do the paralogs show unique changes? The TNRC6 paralogs do appear to be mostly functionally redundant. A may have some more functions, but we're not entirely convinced that that's not just due to its higher expression levels. So what happens to transcripts when Argonaut 2 is bound? So this TNRC6 and this Argonaut binding in this RNAi pathway does not always correlate to repression. There could be other things that are happening, which we found to be really interesting. And then the next thing is, do changes in TNRC6 and Argonaut mirror one another? They do. TNRC6 and Argonaut knockout cell lines have very similar gene expression profiles, again, highlighting that these two proteins are really partners in RNAi, especially when we talk about endogenous RNAi. And the last thing is TNRC6 involved in splicing regulation. And it does appear that some TNRC6 paralogs are involved in a few endogenous alternative splicing events. And that's something we really want to explore more because we think it's a really interesting avenue. So I just want to thank everybody for their attention. I want to thank my lab and my university for always being so supportive. And I will take any questions now.
Thank you so much, Samantha. That was a lot of great work. I will start with one very quick question. So your triple knockout cell line, specifically for the C, so you're using an siRNA to do it. I was wondering, like, how long are your experiments when you use that cell line, and like, is the knockout sufficient for the length of the experiments, like when you do the measurements?
Yeah, so we normally sort of do a double transfection. So we'll knock out TNRC6, and then we would transfect the other siRNA within a couple of days. The siRNA knockdown tends to last about 5 days, and we always would check the knockdown efficiency after we harvested our samples, and it was always between like 95 and 99% knockdown of TNRC6C. It was a pool of siRNAs that really did the job very efficiently. So yeah, we were always very careful to make sure that we knew TNRC6 was out in any sample that we were using for these experiments and looking to publish. So yeah. Sounds
good. Thank you. We have a question in the chat from Catherine, and she's saying that since TNRC2 acts like a scaffold, is it possible that your eClip readings, you can pull multiple AGOs for one binding event and then maybe confuse which transcript is bound to it?
Yeah, so we have some data using a antibody that's sort of for all Argonauts, so it can pull down all of them. And then we have this same data for specifically Argonaut 1 and Argonaut 2. Those are the only two antibodies that are really good enough to do eClip. So we are trying to sort of sort through what, if there's specific transcripts bound to specific Argonaut proteins. Unfortunately, the sort of cross-linking things, we can't really get a lot of TNRC6 because it's sort of a past interaction. We haven't been able to sort of pull that down and then get any transcripts or anything like that because it doesn't directly bind to the RNA. But that'd be really great if we could. But we're sort of using eClip to see if there's different transcripts in the Argonaut proteins. I hope that answers your question.
Roland, any questions?
Yeah, I have two different questions. So the first one is about the gene expression changes. Again, you did all the experiments you did in one particular cell line. So I was just wondering, is there something about, do you know something about these TNRC6 A, B, and C levels in different cell types? Because it might very well be that the one is way more expressed in cell type A, and that the other form is way more expressed in cell type B. So that might also affect the downstream effect of binding.
Yes. So we haven't directly looked at other cell lines. We've sort of been focusing on our model cell line. But if you sort of look at data online, it sort of tends to match that TNRC6A and B are the highest expressed. Sometimes B is higher, sometimes A is higher, and then C is almost very, very lowly expressed. It's almost not expressed or not detected in a lot of cases, or sort of like TNRC6A and B have like 30,000 and C is like at 300, sort of that ratio. So we haven't looked at like a wide variety of cell lines, but we are interested in sort of events that are perhaps more gonna have more RNAi required. So like stress events, or like cell lines or cell types that use more RNAi generally. And so we're going to be looking at like TNRC6 levels and sort of those cell lines or those stressor events and things like that. So yeah, it definitely could change based on the cell line, and we expect it would. Sort of HCT116 is just a good model cell line, so we're trying to get the basis with that, and then hopefully expand out with future projects.
Yeah, and the second part is my interest is the splicing events that happen. So first of all, do you think it's like a direct effect, or do you think it's an indirect effect of all the other gene expression changes that happen, that you just, because that's what you always see, right? If there are gene expression changes, you can also see changes in splicing. So it might not be, or what is the proposed mechanism to let it be a direct effect?
So I think first of all, I think it's happening very rarely. It's not like a super widespread thing, but I think the sort of potential for the direct effect is there, because you can have sort of Argonaut bound to a microRNA, and that happens to bind to an intron that's being transcribed, or an exon that's being transcribed of an mRNA. And then that is then spliced differently because the presence of that protein there interrupts other splicing binding, like the spliceoform forming, or sort of the invasion junction to the A nucleotide that occurs for splicing. So I definitely think the potential is there for a direct effect, but I also do see that it could be an indirect effect, just, TNRC6 is nearby and it just so happens to bind there. But because we did find some microRNA and intron eclip in the nucleus, we do think it could have the potential to be direct effect. We didn't find that many because introns are really transient. They don't last very long. So I think it could be either, honestly. I do sort of lean a little bit more towards direct effect because we did the transfection experiment and did find that we could influence the splicing by transfecting a double-stranded RNA that targeted that site where we found Argonaut 2 to be bound. So we'd like to expand that, test some more loci, and really sort of look into the mechanism of, is it direct, is it indirect, what exactly is happening?
Yeah,
so yeah, that's definitely something we want to explore.
Yeah, I think now you're more like studying the function of this complex, right? So, but do you see any therapeutic applications for these splice events, or do you think you can also do this targeted?
Yeah, I mean, I feel like if there's diseases that are caused by missplicing, we might be able to influence them to splice back properly by transfecting a small RNA. The ones we use are very similar to siRNAs. That's already a platform that's been used for therapeutics. So I definitely think it has the potential, and sort of understanding how it works with TNRC6 and Argonaut could help influence design. Because if you understand the mechanism really well, it's a lot easier to design therapeutics. So that's sort of the reason why we're doing this basic research, is sort of build a foundation, and then hopefully it can be used to help people.
Yeah, fully agree.
So yeah. I would just have to know what diseases. I don't know enough of them.
Thank you for the clear presentation.
All right, thank you so much. I appreciate your questions.
Thank you, Samantha.
Stop my screen sharing.
So now we will move to our last talk for today, which is going to be presented by Catherine Monopoly from UMass Medical School, who is going to be talking to us about the methods to apply and evaluate machine learning models on limited biological data sets through the lens of siRNA design. So Catherine, the floor is yours. You can share your screen.
Thank you. All right.
Perfect.
Let me just move this. Oh, goodness. Okay. So as you all know, I'm Catherine Monopoly, and I am a student in Anastasia Karova's lab at UMass Medical School, and I'm co-advised by Dimitri Korkin at WPI. And today I'm going to discuss some ways that we can overcome the challenges we might face when developing machine learning models when applying them to biological data. And I'll be using siRNA design as a sort of case study. So Samantha already covered this beautifully, so I'll just touch briefly on the function of siRNAs, specifically to targeted gene knockdown. So small interfering RNAs, or siRNAs, are small, approximately 20 nucleotide double-stranded RNAs that can be added to cells. And once inside the cell, they can be loaded into a complex containing Argonaut. And one of the strands of the siRNA is cleaved, and the RISC complex or the mature RNA-induced silencing complex, or RISC, is formed with a single strand of the siRNA. The RISC complex can then bind to target mRNAs through complementary base pairing, and Argonaut can then cleave the mRNA, and the mRNA is degraded in the cytoplasm, and the RISC is then recycled. So this process is highly potent and specific, and that makes siRNAs really useful for therapeutics and research tools. Unfortunately, the efficiency of this process varies for different siRNA sequences, and chemical modifications on the siRNA that we need to enable efficient cellular uptake and stability further limit the sequence space for design, and it makes it really challenging to select effective siRNAs. So to enable more efficient siRNA design, we'd like to build an algorithm or method that takes a target mRNA as its input, performs some calculations, and outputs siRNA sequences and their predicted efficacies for the given mRNA target. The component of the algorithm that performs this calculation is the model. And these models can be very simple, such as a linear expression shown here, or they can be highly complicated, high-order expressions. Machine learning methods allow us to build these models in an automated way using a given data set. So as I show here, machine learning can perform an iterative fitting to identify the optimal model for our data. And this specific type of machine learning describes supervised learning. But to build this model, we first need to identify our training data. So in this example, we will be using a panel of 356 siRNAs with known efficacies. And each siRNA was designed to target a single gene with a total of 17 total genes targeted by the panel. And for each data point, we have an siRNA sequence, as well as its corresponding efficacy with target gene knockdown. And as you can see here in the data set, we have a wide range of efficacies of our siRNAs, ranging from highly potent and therefore very low target gene expression percentages to siRNAs that aren't functional at all with very high target gene expression percentages. But to begin working with these data, we have to consider the type of model we want to fit. So supervised machine learning models can be divided into two groups, regression and classification. A regression model seeks to fit a mathematical expression to the data such that a model predicts a numeric value. So this is pretty typical if you've ever worked with linear regression. Whereas classification seeks to partition the data and predicts a label. When considering our data set, at first glance, regression might seem like the best model, as there's a region here in the middle where the data actually look fairly linear. However, when we consider the entire data set, the model is no longer linear and it becomes fairly complex. Further, when we consider that the goal of our model is to predict siRNA efficacies as either functional or not, this is essentially a label, and therefore a classification model is better suited. With classification models, it's beneficial to have data that are already distinctly in discrete groups. Unfortunately, the spread in the range of siRNA efficacies is fairly continuous in this data set, and therefore there's no obvious place to divide the data. Biologically, however, we typically identify siRNAs with target gene expressions values less than 25% as functional. And we can thus label the remaining siRNAs as non-functional. And I show them partitioned, the functional group is in yellow, the non-functional group is in light blue. We run into a problem, however, when we consider the data points around the threshold boundary. There is a region where the noise overlaps in the data, and thus it is not possible to distinguish siRNAs close to this threshold, making the classification inaccurate, because essentially we have siRNAs on this side of the threshold being essentially the same as the siRNAs closely located on the opposite side. So to alleviate this challenge, we define a second threshold. And this kind of eliminates the noise overlap. However, it creates an additional problem as we now define a third class of siRNAs as undefined. And this is the class that's neither functional nor non-functional. And these siRNAs have to actually be excluded from model training. And this limits the amount of data that we can use. And for any model fitting, it's really important to have as much data as possible, but particularly with machine learning, normally we need hundreds and even thousands of data points to build a predictive model. One way to address this is to adjust the thresholds, which will increase the amount of data available for training. But which thresholds to select is not a simple question. Increasing the functional threshold will increase the number of siRNAs in the functional dataset, but this will also impact our model's predictive power on new data. Less restrictive cutoffs like these may lead to a model that incorrectly predicts more siRNAs to be functional that are in fact not, or false positives. Similarly, a less restrictive non-functional threshold may produce a model that incorrectly identifies functional siRNAs as non-functional. This creates a trade-off between dataset size and model predictive power. To best identify optimal cutoffs, we can consider a range of thresholds for both functional and non-functional cutoffs in every combination of each. And this is fairly easy to do using machine learning since model generation is automated. But with all this concern about classifying your input data, we still have not discussed one very important component, what these data actually represent and how they can be used to build a model that actually predicts siRNA efficacy. So if you recall earlier, I mentioned each of these bars represents an siRNA, and we know the sequences of those siRNAs. So when loaded into RISC, the siRNA interacts with the mRNA through a target sequence. And we've shown previously that this sequence affects siRNA potency and can serve as a predictor of siRNA potency, and therefore we can use it in our model. But we must first convert this sequence into a form that the computational machine learning algorithm can interpret, a matrix of ones and zeros. And this will be performed for all of the siRNAs in our data set. So now we know what our data are, but how do we perform the machine learning and model building process? So for model building, there's a fairly standard pipeline. We first randomly partition the data into a training set and a testing set. We then train the data, we then use the training data to train a model using machine learning methods. Then the testing data are used to evaluate the model's performance. But there's a problem. Because our data set's so small, the data partitioning step is likely to bias our model or its performance assessment, because there are likely to be characteristics in the training data that might not be present in our test data set, or vice versa. And this can lead to a very inaccurate model. So to work around this, we can instead use kind of a different method. So since we have limited input data, we want to ensure each member of the training set has been used in the test set to evaluate our model. So we instead start by partitioning our data into 10 groups. We still hold out the 10% test set and train on the 90% of the data and evaluate as before. But we don't just stop at a single model. We then train a second model using a different 10% test data set and the remaining 90% of the data for training. We repeat this process 10 times, one for each of the random partitions we made at the beginning. And this process is called k-fold cross-validation, or in this case, it would be 10-fold because the k is equal to 10. This is used frequently to minimize overfitting of the model to training data. And it's particularly useful for smaller data sets, like in this example, because as you get to actually larger data sets, the training process can take a lot longer. So this is really beneficial for smaller data sets and not so great for larger ones. So now we can build our final model using the entire data set. And this final model is actually going to be the same one that we ultimately use in our siRNA design algorithm. So through this process, I've been fairly hand-wavy about the specifics of machine learning because most supervised machine learning methods can be applied to the framework presented here. Here I show 3 of the more common supervised machine learning methods used for classification. And I'm not going to delve into this topic, but I'm happy to discuss this with anybody offline. But for the siRNA design example we present here, we're going to use random forest models because we've tested them extensively and they work really well for this data set. So now that our model has been trained, it must be assessed. So for model fitting, we're considering a broad range of threshold combinations. And this can lead to a highly imbalanced data set if, for example, both functional and non-functional thresholds were set to 15%, like shown here. In this case, many more siRNAs will be classified as non-functional than functional. And for such an imbalanced data set, precision and recall are useful metrics for model performance. Precision is calculated thusly and is a proportion of siRNAs predicted to be functional that are actually functional. Recall is a proportion of siRNAs that are actually functional that are predicted correctly. Recall is sometimes called the true positive rate. We can plot the precision versus the recall for different thresholds used in evaluation. And we identify good models as those with high precision with increasing recall. So the solid line shown here, whereas the worser model is the dotted line shown here. The area under the curve is also typically larger for the better model. When investigating models, it's also useful to use contingency tables, sometimes called confusion matrices. These tables can be used to assess the performance of a classification model by comparing the quantities of predicted values with those of the actual values. So using these tables, it's simple to identify the number and proportion of true positives, true negatives, and false negatives outputted by a model. Since we're exploring a large range of both functional and non-functional thresholds, to better organize the results, I'll plot them as a matrix with functional thresholds indicated along the top and non-functional thresholds indicated along the left side. So when we look at the precision recall curves for each combination of functional and non-functional thresholds, we see a pattern of overfitting as the functional thresholds increase. An opposite pattern of underfitting is seen as the non-functional threshold decreases. Looking more closely at this model, we see that our underfit model is predicting all of the siRNAs to be non-functional. And I'll include here a contingency table just so that you can actually follow along with the results if you're unfamiliar with this type of output. And this kind of makes sense that that this model is going to predict everything to be non-functional, because if we actually look at what these cutoffs represent, they represent this entire group overlaid with that. So that means that the majority of our data is being classified as non-functional when it goes into the model. So our model is going to be really good at identifying non-functional siRNAs. And that's about all it will be good at. Conversely, the overfit model essentially predicts almost all of the siRNAs to be functional. And this is because the thresholds here lead to the majority of the training data being classified as functional. We identify the best model as one that balances the overfitting and underfitting, and selected a curve corresponding with the 22% threshold cutoff for the functional group and 65% threshold cutoff for the non-functional group. Here, the contingency table shows that we have a lot of true positives and true negatives, which we want, as these are the correct predictions. So those would be the ones shown here in green and in blue. But we also have false positives and false negatives. But of note is that we seek in our model to minimize false positives, because we don't want a model that predicts an siRNA to be positive if it doesn't work, because it's quite tedious to synthesize an siRNA in the first place and it's expensive. So we want to minimize the number of siRNAs we actually select to produce in the first place. But it's okay that we have higher false negatives. And that's because the ultimate goal of an siRNA design algorithm is to just identify a few or even just one siRNA that knocks down a particular transcript. So we don't need to identify all of the functional siRNAs. And keeping these kinds of things in mind as you develop these types of prediction models is important, because sometimes we can use the wrong metrics to assess our results, and we might have a model that actually works quite well. But if you were to look at the outputs, you would say, oh, there's a lot of false negatives. This might not be the best model, but that's in fact completely fine in this case. And these are just the cutoffs that we selected with our best model. And these thresholds are fairly stringent, and the functional threshold actually still falls really close to the typical cutoff used by biologists to select functional siRNAs. So it's around 22%. And that's important to keep in mind. So normally biologists, as I mentioned in the beginning, identify around 25% or maybe even 30% as the functional cutoff for siRNAs when they're expressed as a percentage of gene knockdown. And we always want to look back at what the model is suggesting to just make sure that it's not fitting to something that seems completely really odd, because we want to make sure that we always are considering the biology. So here we present a method to develop a predictive model despite a small and noisy data set. And we present this hopefully kind of as a framework that we can actually, one could actually apply to other similar problems where data sets are small and noisy. This is really common in biology. And this, I hope, to encourage kind of the application of these powerful machine learning models to data sets that we used to think maybe were too small for this type of analysis. But yeah, and with that, I'd like to thank obviously all of you and for being the OTS, just for being invited here to speak, and both the Kurova lab and the Corgan lab. But yeah. And I'll take your questions.
Thank you so much, Katie. This was a great presentation, especially that, like, at least for me, was not expert. I understand the majority of your work. I have a very quick question. So after the algorithm spits out like this, a couple of like candidates that do you usually like when you test those candidates, do you usually get those data and you feed them back into your algorithm to double check like what was predicted was a true positive and true negative, like from experimental data, or no?
I have actually not, we don't regularly do that, to be completely honest. Normally, so Hassan is actually in our lab, so we regularly, I will predict things, so he knows that actually the algorithm that we designed outputs a score, and it's very specific just to that specific transcript you're knocking down. So it's hard to compare it with the remaining algorithm, but it would be interesting to go back and take a look.
Yeah, it would be, I would imagine it would be interesting, like to see like how many positives and negatives that you got from that set, from the set that you produced, and try to feed that back into your algorithm.
Yeah, so actually, to be honest, the way that it is now, so the results of this I have not implemented in our algorithm that we use in the lab. Right now, I think the true positive rate is only maybe 10% or maybe 20%. The goal is to design 10 siRNAs and to have at least 2 of them work. Yeah. But that's a lot better than it used to be. We used to have to look at almost the entire sequence of the transcript. So it's getting better.
Yeah, we have a question in the chat from Samantha. She applauds you for your amazing explanation of machine learning, and she mentioned that in their lab they found that siRNA pools within 2 to 4 siRNAs combined works best for efficiency. Is that something that could be added, or that you can add to the modeling output?
Yeah, so so you're talking about kind of mixing multiple siRNAs together.
I believe so.
Yeah, that would be really cool. That's actually, so kind of my goal and why I present this is that we're actually using, so it used to be that we would just use linear regression, and linear regression for this type of prediction model really only was amenable to sequence. But now with more advanced machine learning methods like random forest, which is what I'm using here, you can add these additional kind of complications to your experiments, and you can use them as predictive tools. So we could see like how maybe we wouldn't be able to do this prediction now because we obviously need the data to do this, but I could definitely see incorporating kind of this as these additional sequences as features. And it would be kind of interesting to see how that would work. We're definitely considering additional things like other proteins that might be present in the cell as well, which is why actually Samantha's work was really interesting for me.
Thank you. Ronald, do you have any questions?
Yeah, great overview of the work and a very exciting experiments. Also for me, a completely new world. So very exciting. And so my first question is, and because you did a lot of very good and very nice work. So are these kind of tools, are these publicly available? Because that might help a lot of researchers, and very often these tools are for a company or for one lab. So sharing them might have an impact on the entire research community. So
yeah, that's super encouraging. That's my ultimate thesis project. It's a lot of work because to share them, I actually have to build the framework and the website and make sure it's very robust so that I can share this, because running these machine learning models actually isn't trivial to do online, but it's definitely a plan. Currently, we are happy at the Korova Lab. We're happy to work with others. Like if you send me a sequence, I'm happy to predict siRNAs for you, at least the sequences right now. But hopefully soon we will have this published and online within the next couple of years.
And then my additional question is, because you can only help other labs if this method is also validated for other labs, right? Probably
Yes.
you use one particular method for designing and testing the siRNAs, but have you tested this on a completely different lab, on siRNAs from another lab, whether this output is also valuable?
Yes. So actually what's interesting is I used to work in industry before coming here. And I kind of was working on a similar project in both places. And we used not only a different evaluation method, different chemical scaffolds as well. And this method seems to work no matter what. One thing that is true, that sometimes we find that you may need to, as a lab or as a company especially, provide your own data set to train the model yourself so that it works in a specific way. But actually that's not that hard to do. But yeah, even if you don't do that, in general, the types of things that we pick up with these models seems to be applicable across different ways to validate them and different chemical scaffolds.
That's cool. Thank you.
Thank you so much, Katie. Okay, so we are at the conclusion of today's webinar. I just wanted to thank again our speakers. And I wanted to highlight two things. I just wanted to mention that the OTS is accepting applications for board of directors. The elections are being where elections will be held. The applications are due by November 8th. So, and another important thing, which is mostly interesting for trainees, is that for students and postdocs, OTS is also seeking members to join as trainee representatives, and that will be for the year of 2022-2023. And these trainee reps will be like the voice of the trainee scientists who will help organize webinars such as this and many other events that involve the society. So the applications for this are due November 15th. So if you're interested, please check the website. And again, I would like to thank our speakers for today, and thank you all for attending. All right, awesome. See you, everyone.
Thank you, Hassan. It's a lot of fun to join the OTS junior board. That's what I wanted to say.
All right, thank you, everyone.